Okay, I try to hurry up from here on now since we're already a little bit late, but try not to bite too much into a coffee break. So thanks very much, first of all, the organizers for inviting me to come over here and tell you a little bit about the work we're doing. As you see in the title, I continue to some extent what Richard von Jones so beautifully has shown us about before, namely the spatial aspect of some of the signaling that's going on in the cell at the subcellular level here in particular. We'll hear about other scales probably later on. But even if you look at the single cell, there are several scales going up from the protein level to the whole cell dimension. And there's a lot of going on there, in particular with respect to calcium signaling. And we see that if you look at the ultrastructure of the cell, which we think we know about already for many years. There are sketches based on electron microscopy, and there's lots of detail there. There are lots of small compartments in there. And I'm going to focus on calcium signaling aspects of that, where we heard a little bit more about the proton signaling before. And as we know now, they're closely linked, but here I entirely focus on the calcium signaling aspect. And one of the calcium signaling elements in particular I want to focus on is carried by a number of receptors that we call the uranidine receptors, or for short RYRs, which are the intracellular calcium release channels within the cell. And we know they are organized into clusters that probably have 10 to a few hundred of these receptors in a very small nanoscopic, as we might call it now, environment that only spans a few tens of nanometers. And we not only have one or two of these clusters within each cell, but tens of thousands of these clusters. And under normal circumstances, the signaling at each of these clusters is more or less independent of each other under tight control of the extracellular membrane, where we have voltage-sensitive calcium channels. Once they're activated, they squirt out some of the calcium from the extracellular space, and via calcium-induced calcium release, activate these RYRs, ryanodine receptors, or SR calcium release channels to release most of the calcium from the SR and activate contraction. That's a nice, robust system that normally works very well in the heart, but we do know that there are circumstances where we see a more pathological type reaction, where we see propagating waves that might be related to arrhythmias in the heart. And that's one of the aspects we try to understand. And that's where the spatial aspect comes in, when we try to label now these ryanodine receptors in single cells, and we find the typical distribution of most proteins that are of relevance in calcium and other signaling in the heart, with the typical sarcomeric arrangement you see here in the confocal image, where these ryanodine receptors have been stained with antibodies that attach to these ryanodine receptors. And we do know now these things are at what we call the Z lines, the ends of the sarcomeres, and then we see this sarcomeric pattern. Now, if you look into the cell, then we can see these in action. The cell has been loaded with the fluorescent indicator, fluor4, and you see these individual local flashes which reflect what we call calcium sparks, or the local calcium release at each of these clusters. And you see that they're spontaneously occurring, but then there are also these propagating events where the probability of these local signals add up and couple to each other to cause calcium waves. And that is also then summarized here in a more traditional line scan view in one line through this movie. That is a one cell under the confocal microscope at a fairly high time resolution. And you see these bands here of elevated fluorescence, or calcium in this sense, where we have calcium waves sweeping through the myocyte. And we're trying to understand how this transition might occur from the normal robust behavior to this regenerative behavior. And the thing we need to know for that is the distribution of these ryanodine receptors within the cell. This is now a cross-section of a single myocyte, spanning maybe about 20 millimeters or so across. And we can see very nicely in there now these red spots, ryanodine receptor labeling, that shows us where these clusters are distributed all across the cross-section of the myocyte at the level of the Z line. Since we know we are at the Z line here, we've stained another protein, alpha-actinin, that anchors the thin filament at the ends of the sarcomeres. So what you see in green here is the cross-section of the contractile bundles that are going all throughout the cell. You also notice these very large void areas here, and you probably are aware that's where we find it mostly jam-packed with mitochondria and other parts of the cellular machinery. They probably also take a part, but as Richard was alluding to, we might not understand it as well as some of the other signaling. So this looks all very nice. We can describe the distribution of the contractile sites, of the release sites, that might give rise to, say, a wave where we have, again, diffusion potentially coupling release from one side to the next. and it looks like it's a nice two-dimensional distribution across the z-disk. However, if you look at the signals, really, this distribution in 3D, you see it's way more complex than that. So we take this raw data, which is actually from a 3D stack, and we can find quite nicely where the z-lines are, the actual signals, the optical signals, the red ones are quite distributed, but we find the centroids, and we can produce very nice z-line distributions that show us exactly where the sarcomeres end. We can also take the distribution of the RAN receptor antibody labeling signals and try to detect where we think individual clusters are located and replace them with these sort of white blobs that show us quite nicely where these are located. And from this, we can produce a 3D distribution of this, and you get a glimpse of that. It's quite, you see still the sarcomeric arrangement, but it's quite a complex arrangement that is way more complex than the typical planar geometry you might assume, where we have one z-disk, the next z-disk, and the white blobs here that show us the RYR clusters or release sites throughout the cell. It's pretty hard to take this in since these images are always very, very busy. So I'll just show you one aspect of this by flicking to the next one. But I'm looking at one area here in the myocyte. Again, in cross-section, we look at in depth here about 4 sarcomeres into the myocyte. And if I look at this, first of all, side on here on the left, again, this is this red area I showed you before. You see again the contactor machinery here in cross-section, and then the RYR clusters or release sites around that, releasing that calcium that will then activate the contactor machinery and maybe during a wave allow the calcium to spread to the neighboring side and activate those. But if you look then from the top, from the above the cell, the long axis of the cell goes this way, we see the 3D arrangement of the z-lines themselves is in certain cases way more complex than you might appreciate. It's not just planar. If you look at this topology in 3D, it's a zigzag pattern, or you could even describe it as a spiral going through the cell. And that would provide, for example, a pathway of calcium to spread not only from one z-line to the next, but along these connecting pathways along the cell. So we can take such a distribution of release sites and talk to modelers, since we are mostly concerned here with getting the quantitative information, and ask them how would such a complex, realistic distribution of release sites of RYR clusters compared to a planar geometry if we assume what we know about the calcium signaling within each site and the diffusion of calcium from one side to the next and the calcium sensitive behavior of individual sites. And what we can do then is we can compare what we call the realistic geometry to a planar geometry, which has mostly in the past assumed to characterize the geometry of the heart cell very well. We can then initiate waves in such two geometries and look at their relative propagation. You see there, it's not easy to see, but certainly you can see differences. And they become a little bit clearer when you look again at a line scan image that has been computed from such overviews that shows here time and space on this axis. That shows us here now the spread of a wave. In the realistic geometry, we get a fairly robust, relatively fast, typical velocities around 100 micrometers per second or so area of the wave in the realistic geometry, where in some sense the release sites are more isotropically distributed than the purely planar idealized geometry. By comparison, in the planar geometry, we get sometimes partly abortive wave propagation much more slowly than that. And the bottom line here is that regardless which is the right distribution, the spatial distribution certainly is a very important influence on how the system behaves. And you have to think about it if you want to develop a proper understanding of the system. This was just a system having a few sarcomeres, and our collaborators Aaron Holden and Pen Lee have carried this further and taken such a distribution and created a whole cell model, which is computationally still quite costly, and looked at wave propagation now in this fully extended realistic cell model versus a planar geometry, and there are clearly still large differences between these two geometries where we find more isotropic wave spread in the realistic geometry. This is all very nice and I think shows some potential of this approach, but I want to also point out a shortcoming, and that is we're using optical data. And optical data is always limited in its diffraction limited character, that we can only see things at about 250 to 500 nanometers in cross-section. But actually, the actual data underneath such a normal fluorescence image is a lot smaller. The individual proteins are on the scale of nanometers, and even the clusters are only a few nanometers. So how can we get more size information? Traditionally, we used electron microscopy, but now there's a nice trick that's come about and we've applied, and that is you can look at the fluorescence in a different way. If you take that same labeled cell and put it in the right chemical environment, low oxygen, and put our lasers up, we can then look at molecules, individual molecules, look at individual single molecule fluorescence. The same distribution, the same cell I just looked at, we're now looking at over time, turning our laser up, and we don't see a static image anymore. We see flickering, blinking events. We see each individual molecules of our stain at any one time. And when we use this type of data, we can use it to localize where the individual molecules reside. And if you do that, we find that we can, first of all, see it still with its diffraction-limited diameter, but we can also localize, since we know now each of these molecules reflects just one fluorochrome, where it resides at the center of these flashes. Each of these is 250 nanometers apart in diameter, but the center we can now localize to 20 or 30 nanometers or so. And that gives us a lot more information. So first of all, if we do that kind of thing, this is the original image. We take all our single molecule events and we can rebuild from just summing up the single molecule events, the diffraction-limited picture. But we can also have what I call, if you know this kind of TV series, the CSI event, where we get a lot more information, a lot more spatial information. And I'll show you that in a bit more detail. We have all these single molecule events. Now this is a 100 nanometer scale. And each of these little colored blobs is one of these localized single molecule events where we think a fluorochrome resides, much higher resolution than typical diffraction-limited images. This is the corresponding diffraction-limited image. I put on the CSI effect, and this is what we see again when we use this high resolution, the localized fluorochromes. And we can now see that each of these little patches has detailed structure that reflects the individual RI clusters. We can see small clusters that were not at all apparent. I'll show you again an overview that compares here in peripheral RIR clusters. The red signal is the traditional diffraction-limited image. The green data is the high-resolution single-molecule localization data, and we see lots of small things that we didn't pick up with the low diffraction-limited data. In fact, you can carry it so far that you get near single protein resolution with this type of data. This is the actual data that we record for one or two of these clusters, and we can contrast it to the size of an individual RYR, which is about 30 nanometers across, and they're supposed to range in these very regular arrays, and we can see we can one by one more or less match our actual data with the receptor distribution and count how many proteins are within these clusters. And we get a lot more information that we can now start to feed into models. There's a huge variation in shapes of clusters. It's just an overview of different cluster shapes we get. We can do morphological analysis of that. For example, we can look at the aspect ratios of these clusters. Previously, they have been assumed to be more or less circular. We see that the vast majority is elongated. So many of the assumptions we get from thin sectional electron microscopy don't seem to be particularly justified. So based on that quick overview that can only give you a glimpse of what we think is possible in a program that we're hoping to carry forward, we think fits very well with the ideas of physiome. Quick conclusions. We think that detailed data is critical for proper model construction and basic understanding what aspects of that spatial distribution are important to our understanding. We think there's a great demand for good tools to ease incorporating this data into models. And in particular, it would be nice to have a standard for exchanging image data that is at different levels, at raw, segmented levels, or further refined levels into model construction, so the exchange of that data with modelers is eased. Maybe there is already something you will tell me. I'm not too aware of it. And finally, I think there's great excitement now about these new fluorescence methods, at least for me, that we can use the great contrast in fluorescence methods, get really high resolution that starts to resemble that of electron microscopy and start to complement the electron microscopy data that is now being able to use tomography to also give us exciting new insight. Finally, then to conclude things, my collaborators, the confocal data, worked on together with Mark Cannell, who was leading this project with me, Isuri Yarasinghe and Shari Kong, who did some of the imaging. The localization high resolution material spearheaded by David Bedley, a postdoctoral fellow who was instrumental, and Usuru, Leo, and Sabrina, who worked on the chemistry and the imaging aspects. Simulations from Aaron Holden, University of Leeds, and Pan Lee, who is now in Norway at Simula. Localization work together with Christoph Kramer from Heidelberg, and funding from various New Zealand bodies, as well as the Wellcome Trust here in the UK. Thanks very much for your attention.
Okay, we have time for one short question. Well, it looks as though everybody's desperate for coffee.
One question is, can you put this into perspective of cellular hypertrophy and how new sarcomeres are laid down at the ends of the cell if you have this spiral arrangement?
Yeah, it's. I guess one question we have is how does it arise, this type of spiralation? First thing is that Lee Peachy saw something like that in skeletal muscle probably 20, 30 years ago. So it's not unique to cardiac muscle. And if these type of spiraling or other geometrical irregularities gets more pronounced in hypertrophy, is still an open question. We certainly have some data that we think even under load, and it's in a tissue context, they still remain, and they might have to do with additional sarcomeres being laid down and different stresses, so that I guess you can match two sarcomeres that have slightly different number of sarcomeres into the same cell.
Right, thanks, Christian. I'm going to stop things here. So we're running 5 minutes late, but can we still reconvene back in here for the second session this morning at 11?